Hello, everyone. Welcome and thank you for joining our webinar, How a Publicly Traded Gene Therapy Company Optimized Construct Designs with FORMsightAI. My name is Angela Anderson, and I'm the Senior Director of Marketing at FORMBio, a life science-focused software company dedicated to empowering scientific discovery and enabling a new era of insights and breakthroughs. Recently, FORMBio was selected as the exclusive bioinformatics platform for Google's new multi-omics suite, and we've partnered with PacBio for streamlined cell and gene therapy solutions. So there's a lot of really exciting new developments. Today, we will be discussing how AI can help predict and optimize construct design for real-world gene therapy manufacturing. And I'm thrilled to introduce our FORMBio speakers, Andrew Busey, co-CEO and co-founder, as well as Dr. Claire Aldridge, Chief Strategy Officer. You can expect a 30-minute overview of the state of the cell and gene therapy industry, followed by real-world case study on how gene therapy company leveraged AI to streamline their development process and bring new treatments to market. And also we'll be conducting a short Q&A at the end of the session. So if you have any questions during the webinar, please type them into the chat window and we'll address them during the Q&A. So without further ado, I will hand it off to Claire. Please take it away.
Thank you, Angela. Thank you for having us. And we're looking forward to telling you about what's going on in cell and gene therapy and how we think this is a perfect application for the AI tools that have been developed over the last 18 months and are really accelerating discovery all over the place. One thing at FORMBio that we're especially excited about is cell and gene therapy. It's an enormous market, and it's expected to continue growing at 39% over the next few years. And it is believed that it will be, over $40 billion market shortly. So we think that, again, not only is this a place where AI is useful, it's a real need, a real large market opportunity. What we know about cell and gene therapy manufacturing is that this is a product, a manufacturing process that we haven't done a lot of optimization with yet. So you will have bioreactor runs that are not the most efficient and sometimes fail, and that you can lose millions of dollars as well as months of time. The other important thing is that when your biomanufacturing is not as optimized as it could be, you'll end up with capsids filled with things that are not your therapeutic construct and that are hard to purify out with the purification methods that we've got today. And that increases your risk of adverse immune responses, safety signals, and it also reduces the number of doses that come out of each manufacturing run. And the FDA is really starting to look at this more closely. You know, initially they were very interested in full versus empty, but what we're seeing over the last few months is really, let's understand what's in your full, because sometimes that might be non-therapeutic and we need to get a handle on that. We need to understand what that is. And we need to do what we can to reduce the amount of those non-therapeutic capsids that are potentially going into patients. But all of these lead to a prohibitively high cost per dose. Right now, these kind of therapies are used for orphan diseases, rare diseases, places where that cost per dose can be tolerated in the marketplace. But if we're going to use these modalities for larger patient populations, then we need to figure out how we can manufacture these drugs like we did with monoclonal antibodies about 20 years ago so that they can become more broadly adopted. Next slide, please. For those of you who are in the cell and gene manufacturing space, this is known to you. This is a manufacturing process for gene therapy. You have helper plasmids, you have your gene of interest, your and your cell line that helps package it. And these are, really, this is a process that we've been working on improving, but we still have a lot of area to improve. And what we do know now, what we've spent a lot of time with is how do we have a better capsid? How do we get a better helper virus? Things like that. What we think is let's move all of this upstream and let's take that payload and optimize that payload for replication efficiency so that we can have more full therapeutic constructs able to be packaged. Next slide, please. What the industry has been doing is, kind of a baseline optimization. Let's look at the codons. Let's make sure the codons are optimized for humans. Let's also look for CPG islands, trying to reduce the kind of innate immunity and triggering the safety signals. What we've built into our AI model is really looking beyond that. Let's look at more than just those things. Let's start looking at the secondary and tertiary structures. Let's understand what is it about those secondary and tertiary structures that lead to inappropriate truncations that then can lead to homologous recombination with the plasmids and also just reduce the efficiency of the replication. Let's start looking at other areas using different promoters, for example, and swapping out promoters that have similar tissue expression, similar strength, but perhaps within the context of your gene of interest, reduce the propensity for truncation, things like that. So this is where we're going. And next slide, please. And so what we've done is we've built an AI suite of products that allows us to, one, simulate the bioreactor run. We can take your gene of interest, your construct, we can model it in silico so that you can get a sense of what your manufacturing run would be before you even spend the money to do that. We can also use that information and the machine learning that we've been able to do, trained on public and proprietary data sets, optimize that payload for replication efficiency to improve your final product. Next slide, please. And so, just to kind of give you a quick overview on that, nowadays, I think what we know is that we often take something from R&D that showed proof of concept in an animal. We do a clinical manufacturing test run that allows us to make product for our IND enabling work, for our phase 1/2. But what this doesn't allow us to do is to really understand how we can improve this process. We're often just translating kind of an academic or basic process into a GMP manufacturing run. But what we think is incredibly important is to start to think about that biomanufacturing and how can we improve that to make sure we get enough doses for those early clinical trials, for that IND enabling work, and to improve the safety of that product. Next slide, please. So what we're doing is we're taking that bioreactor test run, and what we're working on is trying to figure out how can we model that for our customers so that they can get a sense of what that therapeutic output would be before they go into that bioreactor run using both financial and time resources. Let's do some of that work upstream so that the bioreactor run has a greater likelihood of success. Next slide, please. What I'd like to do now is turn it over to my colleague, Andrew, who's been thinking about the use case for AI and hard software problems for a long time, and what he thinks about as far as taking what we've got today and how do we push this field forward.
Thanks, Claire. So at a high level, as Claire talked through the problems that exist, they map well to kind of the strengths of AI, which are simulation, optimization, and prediction. We've started with the bioreactor simulation phase, which basically simulates a bioreactor. And that has been an interesting and fun AI challenge for us. Probably the biggest challenge, I think there's already a question in the Q&A section about this. One of the biggest challenges in this category is there's a I think belief that there's a lot of data, but the data itself is relatively sparse and comes from different silos. I think combining different data sources, building our own first-party data. We've been doing a lot of wet lab work that I think Claire will talk about later to build these these models that span these three categories that you see. So the bioreactor simulation is effectively a simulator. Optimization is really about understanding the combination of sort of all the many optimization paths for that construct that Claire touched on earlier, and then simulating the outcome of that optimization and comparing it to the baseline that you built with the initial simulation. So over time, you can not only simulate all your different scenarios, you can optimize them and verify that the optimization is more effective than the original starting construct. You can use these parts independently. So you could simulate just your own constructs without using our optimization AI. You can do verification by comparing simulations that you've done on your own constructs. You can use our optimization and compare the outcome of that optimization with the original via simulation. You can compare multiple scenarios. So there's a lot of things that you can do with the combination of AI components that we've built with FORMsight, and we're really excited about bringing that sort of vertical AI solution to the CGT world. So this kind of looks a lot like the previous slide, because we're basically replacing the actual manufacturing process with simulation. So that is, I think, one of the fastest places to benefit from AI in categories that have not yet benefited from this sort of rapid advance in AI over the last couple of years. Again, the biggest challenge here is finding the right data sets, tuning those data sets, and getting the output correct, because there's a myriad of ways that bioreactors can be configured. There's multiple steps over time to this part of the process. We will, in the future, probably be talking a lot about the purification step, which comes right after the bioreactor manufacturing step, and making every step in this process simulatable so that you can both save time and money and get better results. Claire, do you want to talk about the actual
Yeah, absolutely. This is an example of the simulation report. One of the things it does is it maps your construct. You can see that at the bottom, the nucleotide position. It gives you a score of your truncation propensity. What is the likelihood that you will have a truncation event there? And we also map that, we'll get into this a little bit more, to some secondary and tertiary structures that our machine learning has identified as signatures. I like to think of them as like a three-dimensional signature of a sequence that slows down the machinery enough that there's a tendency for the machinery the replication machinery to fall off. Another part of this is we can model in your non-empty capsids. So, pulling out the empty capsids with the purification process is something that we can do based on size. But for the non-empty capsids, that's sometimes more difficult. And again, because we've trained this model on a lot of data, we can tell you of those non-empty capsids, what are they filled with? And so for this particular example, you can see that this particular sequence would give you a truncated product inside your capsid 60% of the time. 28% of the time you would have a full read, and about 12% of the time you would have a chimeric. Again, these truncations often lead to kind of ends of the DNA that can then lead to recombination with the helper plasmids. And that's what we see a lot of. And we've learned a lot from our deep long read sequencing of these kind of actual bioreactor runs that have trained our models. We could also give you a sense of, oops, would you go back just a second, of the, from a time, when do you get the greatest percentage of non-empty capsids to really try to improve, to kind of stop your bioreactor run at the most efficient time to get your best quality product. And then we can also, in those truncated, because we can model that truncation, we can give you a sense of where do those truncations happen, so that, again, you can understand where you can make changes in your sequence if you're able to reduce that truncation propensity. Next slide, please. Andrew, can you talk a little bit about the optimize, the AI behind the optimize?
Yeah, the so optimization is a little bit different than simulation, although it does work a little bit in a similar way. So optimization is about identifying the opportunities to alter the construct to get better outcomes. There's a lot of ways to do that. There's more than any human could iterate through. So it's a combination of prediction and iteration and optimizing those different configurable parts, and then simulating which ones have the most likely best outcomes.
And I think an important point there is doing all of that, keeping the gene of interest constant. So not changing any amino acids in that protein, but using some of the synonymous codons. And also important is keeping the GC ratio of the entire construct within that 49 to 51%, so that you don't skew that away from the what we know about how this becomes expressed, the expression level of that construct. If we know that if we mess with that CG content too much, we can drive down expression, which when you think about gene therapy is a very bad outcome. And then with the verification, what we're able to do is we're able to take that simulation of your original construct, and then we're able to optimize it using all of those things that we are able to change, swapping out promoters again that have similar tissue distribution, similar strengths, using those synonymous codons, other tolerated changes in that DNA code. And then we can compare the two to give you a score of how much can you change your bioreactor output by doing some optimization. And each construct has a different ability to be improved. Some of them we can improve enormous amounts. Other ones we can't improve as much because whether it's the coding region is already pretty optimized just naturally. So we can give you a sense of what that score is. Next slide, please. All right, Andrew, do you want to talk a little bit about this?
Yeah, so the objective is to optimize manufacturing, which I think is the real opportunity there is to save both money and time by getting safer therapeutics to market faster that are more efficiently manufactured. So either cheaper and or lower COGS. The ability to simulate these steps and grow the various phases of manufacturing that can be simulated and then optimized is a core focus of what Form is working on, and we think that will have a significant and material impact on the biotech industry over the coming decade, and we're excited to help people achieve that efficiency.
All right, so now what I'd like to do is just kind of walk through a case study of a partner that we worked on with this. Obviously, when we first start translating things from proof of concept in animal models, a lot of what we've done in the past for these modalities is what I like to call kind of brute force science, right? We just do trial and error, and we don't have the time or the understanding or the resources to do multiple iterations to figure out what the best one is. So sometimes you will end up with low yield or no yield bioreactor runs. And especially for early stage biotech companies in today's financing climate, that can really be devastating for a company. Next slide, please. The problem that came to us was that holding the manufacturing process constant, what they saw was that the drug product was vastly different when the only thing that was different was the payload. And you could have drug products that were very, very replicatable and made a really nice consistent drug product, almost 95% full reads. And then others, again, the only difference being the DNA sequence, 53% full and 47% of truncated. And this is after basic optimization for kind of the basic tools that are out in the marketplace now. as well as after a purification step to take the empty ones out. So that was the problem that was brought to us. Like, how do we think about that? And we felt that this was a problem very, very solvable by using the large language models and the neural networks that have really accelerated over the last 18 months, and that this would be a great use case to look at that DNA sequence and apply machine learning to figure out what would be a better payload that wouldn't change the final drug product, which is the protein that's being delivered to these patients as a gene therapy. Next slide, please. What we saw when we looked and we started modeling and we started learning from the data sets where the truncation happened is that there were secondary and tertiary structures that were that kind of drove truncations, and that there were secondary and tertiary structures that were protective, that kind of reduced the level of truncation. And so our machine learning model has been able to differentiate those two and help identify what is a signature of a DNA sequence that is less likely to truncate versus not truncate. And, this is biology. I think we all know nothing's binary. We're not going to take this down to zero. But how do we nudge it more into the world of this will be a replicatable payload? Next slide, please. So what we actually did is we were able to map those truncation propensity scores to the different structures. And interestingly, there was a handful of them that really seemed to be about 130, 110 nucleotides away from where the truncation happened. So it makes sense that that secondary, tertiary structure leads to something in three-dimensional space that reduces the ability of that DNA to be replicated. Next slide. So again, you see pointing out the four structures that we now know are associated with this. Next slide. So we believe we've identified that three-dimensional signature for both propensity and magnitude of truncations. Next slide. Another thing that we found when we started looking at this is that different promoters will also have truncation propensities that can be very deleterious to the entire construct. And so for this particular drug product, what we were able to do is swap out for another promoter that, both of these promoters are in development for gene therapies. And so what we know is that for this particular DNA sequence, because there is some kind of proximity effects with these things, but that a different promoter reduces the amount of truncations considerably. And to be frank, much of the truncations in this particular construct were driven by the promoter. Next slide, please. And here you can see an example of what we were able to do using the synonymous codons that allow for reducing the truncation propensity. The dashed line is a line that kind of is one of those tipping points where if you are above that line, you have a likelihood of truncation. And you can see that when we optimized this, our little gold dots, we were able to get a number of those nucleotide regions below the line, so reducing the likelihood of truncation. Next slide, please. So what we were able to do with this through our AI modeling is predictively increase full reads by almost 20% and decrease the number of truncated reads by 70%, which I think is incredibly valuable when you think about, the cost of goods for each run is going to be the same, and the number of doses that you can get out of each run, it's very helpful to maximize that. Next slide, please. This is where we are today, but we are continuing to push the field forward. One of the things that we've been working very closely with PacBio with is when you have your construct that you've developed and you've done your PacBio long read sequencing on, we can analyze that long read sequencing and give you a report that will actually outline what your drug product is made of. So you can have a sense of what proportion of my drug product is full reads, what proportion are truncated reads, what proportion are chimerics or host genome. And that allows you to share with the agency some confidence in what your drug product is. And I think we've seen with some of the recent interactions with the agency that this is something they're starting to ask for more. We're also continuing to sequence drug products from academic partners, as well as from some of our commercial partners, to continue to train our model, and then also doing bench experiments to validate all of this work in a wet lab. Here's just an example as we resequenced a construct, and how the construct had initially been run, had been sequenced about 2 years ago. And we were able to actually get a hold of that drug product again, resequence it with higher read, consistency, a deeper read. And what we were able to see is that as we improved the data, that our old models were accurate, that the truncation distributions were very similar, and that this asymmetry was very similar and consistent, but that we're able to improve our confidence intervals in our reads, and to really feel like we're narrowing our error bars for our machine learning models. Next slide. And then finally, I did mention that we're doing a lot of this work at bench scale to take a look at, how is this all, how does it all translate into the real world? Next slide, please. And so what we would love to do, we are, a company that hasn't even been around for quite a year, and we're working with a number of cell and gene therapy partners. We would really like to partner with you. We'd like to, work with you on the problems that you've got. What are the questions that you have in your biomanufacturing runs? Are you interested in a deeper analysis of what's coming out of your bioreactor? Do you want to model your bioreactor runs before you do it? Do you want to figure out, that your drug runs aren't making, a lot of doses per run? We can help you optimize that. We're doing all of this right now in AAV, and we're able to do upstream processing for simulation and optimization. We are starting work to do this in lentiviral vectors as well as LNPs, and then also adding on a purification simulator so we can start to really model that entire biomanufacturing process, so that we can, get these drugs through biomanufacturing and into clinical trials faster, so that we can get patients treated faster. Thank you. Would love to take some questions.
Yeah, we have a few questions from the audience. Let's see. The first question was, which public data set did you use for AI training model?
So I think all three of these questions kind of go together. So let me try to address them. We haven't used any specific public data sets to train the models. Part of the challenge is most of this data is not publicly available. So we've spent a lot of time gathering it through partnerships, through direct relationships, and also doing our own sequencing work to build out a more sort of detailed data set that we can use to train. We are doing back testing with partners to validate that the bioreactor simulator matches what they've seen in real life results. That usually is not public data. So I think that hopefully that answers these.
We have gotten some public data sets from some of the academic labs that are really pushing this field forward. I think Phil Ty being one of them. He has some public data sets that we've been able to take a look at and use as well, on top of the proprietary data sets and the data sets we're generating ourselves.
Okay, thank you. I think to kind of follow up on what you were discussing is, when you say that you are not optimizing the transgene of interest, could you please elaborate on the pros and cons of not optimizing for that transgene?
We will optimize the transgene using codon redundancy in the genetic code. What we're not doing, because of where many of these products are in the development path, we're not changing kind of that fundamental, what is the gene that's going to be, transcribed and then translated from this construct. the farther upstream we can get with our partners in the discovery phase, we can do more work optimizing the transgene around other things like, including introns, taking out introns, other kinds of spacers that you might put in there, regulatory elements, all of that. But for customers who are, really ready to go into IND enabling work, they're not in a place where they want to do a lot of changes to their construct. So really what we do there is look mostly at the codon redundancy, but we have the ability to look at all sorts of different areas. It all depends on where our customer is in their development path.
Okay, great. And we have time for a final question. How do you incorporate variation in bioreactors and purification processes into your bioreactor simulation?
So I'll take a stab at that one, and then Claire can correct me or polish my answer. So first off, we currently separate out bioreactor and purification as two different steps. So right now we don't combine those. Purification is in the early phases of development. Bioreactor simulation is further along. The bioreactor simulation definitely, and purification is even more complex because it has a much broader configuration set. There's a lot of variability across these things. So today, what we typically do, and what I think Claire mentioned on the previous slide about the lighthouse customers, we partner with customers that to emulate their specific bioreactor processes right now as we continuously broaden the overall bioreactor simulator. We don't really let you go just throw whatever you want into it right now. That will probably come in time, but today the bioreactor is to some degree customized for each partner because there are a substantial amount of differences, and the data for each of those variations is not what I would call complete. So that makes it much harder to build a sort of a general all-purpose answer to every problem.
And that, I think, just to kind of add to that, I think Andrew mentions the data as we build out these data sets. But, one of the things we've learned is that, there is a lot of variability in both the quality and quantity of the data. And so that's, that is a rate limiting step that will continue to be addressed as more data is generated and we're able to partner with more folks. But with some of those, we can do a little bit of modification. We can, kind of model, using a different concentration of, your payload DNA. We can model maybe using a different set of helper plasmids, but we're really partnering with people to say, what is your bioreactor process? So we can model it on that process. And then we'll ultimately get to where, let's come up with an optimal bioreactor process for this payload.
Great, thank you. And before we sign off, if you're looking to elevate your CGT manufacturing, again, please reach out to us, scan that QR code. We're eager to connect. I realize we didn't get to everyone's questions. However, I will be following up directly. And thank you all for joining FormBio, and please enjoy the rest of your day.
Thank you for having us.
Thank you very much.